Hello everyone, thank you for waiting. Welcome to IEEE Distinguished Lecture Series organized by IEEE Student Branch, UEM Kolkata. One second. UEM Kolkata. Today our speaker is Dr. Sumit Soman. Dr. Sumit Soman is Senior Data Scientist of Ericsson and he is a Ph.D. from IIT Delhi and in machine learning. He was principal technical officer in Center for Development of Advanced Computing. Today, the topic of our distinguished topic of our webinar is machine learning approaches for biosignals. And again, thank you for waiting due to some technical problems. It was getting late. Thank you for presenting today. Thank you for participating in today's webinar. And sir, we are honored to have you as our distinguished speaker in our webinar, Distinguished Lecture Series 2021. We are honored to have you as our speaker. Thank you for accepting our invitation. And sir, we can. The stage is yours. You can start now. Thank you, sir.
Thanks, Samandar, and hello everyone. I hope you're all able to see and hear clearly. And thanks for the introduction as well. So I'll try to share my screen and just check the presentation first. And let me know if you people are able to see. So I believe you are able to see the screen. Can someone confirm?
Yes, yes, sir.
Yes, sir.
All right. So, welcome everyone. This talk is primarily going to be more around machine learning approaches for biosignals, where we are trying to look at what are biosignals and what are the challenges that we face when working with these biosignals. Most of the talk will primarily look at a couple of signal modalities, which include brain signals and signals of the muscle movement. In addition, we'll be looking at how we should build a signal processing pipelines and machine learning approaches that can be used so that we can build systems that can use these biosignals. I have been introduced, so mostly I am giving this in my capacity as a senior member of the IEEE, and most of the work that I am presenting in this presentation was done at CDAC and at IIT as part of my Ph.D. And more recently, I joined Ericsson as a senior data scientist. So let's step into the talk and start with the outline. How we'll be going through this is I'd like to keep it interactive. So at any point in time, if you have questions, I urge you to put it on chat, or if you could speak out as well, that would be welcome. And of course, towards the end, I'll try to collect all the questions that you may have put in chat and try and provide answers to them. So broadly, how we'll be going through this talk is we'll be looking at biosignals, specifically in the context of what are called as brain-computer interfaces and neural processes. I will talk about what are these in the coming slides. So one of the key ways of acquiring signals in the brain is through electroencephalogram or EEG signals. So we'll be talking about how we can look at EEG signals, what are the various approaches that we can use to detect intent from EEG signals, what challenges we face, what is the processing pipeline that can be involved. And I'll be discussing results from a couple of papers and some of the work that we had published as part of my PhD and some of the work that I had done at CDAC. In addition to EEG signals, we'll also be looking at surface electromyogram or EMG signals. These are also signals that generate from the muscle movements. So these are sort of used in implants and prostheses or prosthetic limbs. And here also we'll look at what kind of challenges we face, what are the processing pipelines, and results on some data sets that are employed in a prosthetic limb scenario. And finally, we'll look at some conclusions and what are the future directions that work in this domain has been taking and where all you could also be involved in. So to start with, we'll just briefly walk over what are biosignals and what are the different types and how these kind of systems work. So at a broad level, biosignals refer to a very large segment of signals. So if you look at this plot on the top right here, you will find that there are various kinds of signals that are shown here. So this involves various space scales and time scales. So when we say time scales, it is essentially the resolution in time at which you are capturing the data. And then it's a space state, it is essentially how close you are to the source of the signal. So basically in terms of physical proximity to the signal neurons. So as you would know that brain has a large number of neurons and the populations of these neurons fire when they execute various actions. So what you collect as a brain signal is essentially a result of the electrical activity due to a firing of a population of neurons. So you can collect it in various ways. You can collect it from outside, like if you're wearing a helmet like this and it is EEG, it is called electroencephalogram. There are various other non-invasive ways shown here. So magnetoencephalogram, functional magnetic resonance imaging. These are all ways that are primarily non-invasive, where you are collecting these signals from the scalp or from outside the brain. So you are not making any incision or any invasive method inside to go inside the brain. So you will find that these signals have good time resolution in some cases, for example, fMRI, but of course you are away from the source of the signal. So basically you are away from where the signals are generated, and that is why in the space axis you will find that these signals are closer to the lower part of the space. And when we go to finer resolution, where we go closer to the neurons and invasive audiography, local field potentials, you have what is called as invasive signals. So invasive approaches, the main advantage is that you get high resolution signals. But of course, the difficult part is that you have to go inside the brain. So it is largely part of medical procedures. So implanting these kind of electrodes or putting microelectrode arrays inside the brain, these are largely done as part of clinical procedures or medical procedures. So when we look at a brain-computer interface system, this is one sort of a biosignal. So basically, we are trying to look at EEG in this presentation. But when we look at EEG signals, they are generated from the brain. You will capture them using some acquisition device that you will wear on the scalp. So if you will have some kind of a helmet which will have various channels, and you will acquire these signals digitally from this device. So it will be hooked up to your laptop or a PC or a server through conventionally a set of wires. But of course, nowadays you have portable devices that allow you connections on Bluetooth dongles and so on. So it can also be a wireless interface, and this data that you get from this acquisition device or a EEG helmet that you are wearing, that will be collected to your system as a digitized file. So you will primarily have samples that are collected at a specific sampling rate along various channels. And once you have this time series kind of data, you can apply some signal processing and feature extraction method to convert them into a representative feature space. And then you will use a translation algorithm, or you will use a classifier or a regressor or a machine learning approach here to essentially try and detect what the user was trying to do. So the broad idea here is that you have signals from the brain, which are EEG signals. You are using some kind of a feature extraction and machine learning pipeline to process these signals and detect intent. And once you have detected what the intent was, you can use it to do various things. So you could use it to, for example, say control a computer or control the movement of a mouse on a computer system. Or you could also use it to control wheelchairs. So digital wheelchairs, you can have movements that are triggered by your actions detected through EEG signals. So you will, for example, imagine movement of front, back, and so on, and you will try to translate it into the actual movement of the wheelchair through actuation from a server-oriented pipeline. So this is one way in which a BCI system can work, and this is driven by EEG signals which are non-invasively acquired. As I said, that there are other signals also from the brain. So basically the brain is essentially the core of the nervous system that is connected to the spinal cord, and then there is a nervous system with a set of nerves that run across your body. So you might have also seen that there are various people who have lost arms or legs or other limbs due to accidents or some other unfortunate event. So even for those cases, their nerve activity, if it is intact in the region around this, so for example, if their hand is amputated, but still they have alive nerves here that still give electrical impulses, there are devices that capture these signals as well. So this is what is called a surface electromyogram. And these signals are used to run a prosthetic arm. So the user will be fitted with a prosthetic arm, and then you have to detect what the user is trying to do with the arm and translate or move the arm accordingly. So you have multiple degrees of freedom, so you can have multiple movements to capture here. But this is one kind of system that works on electromyogram, so surface electromyogram signals, and it will allow you to move the prosthetic limb and control the user's hand despite it being physically amputated or non-functional due to some reason. So in this talk, we look at one set of PCI systems and one set of EMG systems, or this kind of human-computer interaction systems built with EMG and with EEG. And we'll try to understand what are the challenges in each of this case and how we build the signal processing and machine learning pipeline in both of these cases. So I hope we are good till here. And any questions at this point, or anybody has any thoughts, we can briefly pause for any questions. Okay, so I'll continue the first part of the talk where I'll try to talk about what are EEG signals and what are the challenges that we face when we work with EEG signals. And we will look at a specific approach using classifiability and the twin support vector machine that has been used to work with EEG signals. So this is a paper that we had published in Applied Soft Computing. It is available online. So we first begin with what are the challenges that we face when we work with EEG data. So as I mentioned in the first slide, EEG data is usually collected on the scalp. So as it is shown in this figure, the user as the subject is wearing a scalp map or this kind of a wearable device on the head, which has multiple electrodes or digital sensors that are acquiring the signal. So it is capturing brain activity, and the electrodes are placed in a circular region which represents your scalp. Okay. So this is a placement system called as the 10-20 system, where essentially you have 20 positions to place from, and you have these are placed in a ratio where this is 10 to 10, the split of the ratio. So various regions of the brain have various functions, and accordingly the electrode positions are named as well. So for example, the F electrodes are the frontal electrodes. These are the set of frontal electrodes. Then there is a cortical region which is responsible for motor movement and imagination. So this is the cortical region, has 3 electrodes. Then there is a parietal lobe which is responsible for other functions, which has 3 electrodes. There is an occipital lobe in the back which represents your vision and occipital nerve functions. So this has a couple of electrodes here. Then there are temporal electrodes that are placed on both sides of this. So depending on the region of the brain, the electrode position may vary, and accordingly 10-20 system of electrode placement is used for placing these electrodes. So this is a standard system, and all devices that acquire EEG data generally tend to follow either the 10-20 system, or there are also other variants of this, but largely based upon this kind of a placement of electrodes. So the basic idea of doing it is that you will be able to find out which region of the brain you are collecting signals from. And if you are interested in that kind of function, you can always look at signals from that specific area. So for example, if you are trying to look at some functionality related to eye movement, if you are blinking your eyes or you are closing your eyes, so that is generally captured in the occipital region. So you will try to put electrodes here and capture these signals. On the other hand, if you are trying to look at some movement of the limb, so for example, some motor movements where you are trying to raise your hand, move your legs or something like that, you will look at the cortical region. So you have left motor cortex, right motor cortex, and accordingly you can place electrodes in this region and collect data. Okay. So this is where it is the importance of the 10-20 system of placement of electrodes, and according to this map, you will be collecting the EEG signals. Now, usually these signals are collected from some sort of an acquisition device. So an acquisition device is essentially a set of sensors which collects the data as a time series data. So digitized data is collected from n channels. So for example, if you have used 5 channels in this region, then you have 5 channels. It is a common terminology for EEG data. And you have some sampling rate. So typically, let us say your device samples is 128 Hz or 256 Hz. So you are collecting those many samples from each channel. So from each location that you have placed an electrode on, you are collecting those many number of samples. And you are typically collecting it for a duration T. So how T is determined, for example, if you are trying to build a system to move a wheelchair. So you would want to say move it in 4 directions. You would want to move it front, back, left, right, and you would have one position to stop. Now, every time the user will want to move it forward, you will want to detect the intent from the EEG signal. So let us say you want to detect it every 2 seconds. So you will keep collecting data in a continuous stream and you will window it for every 2 seconds, right? Whenever you want to detect an intent for whatever time interval you pick, that many time windows of data you will collect. So essentially, if you are looking at data from a specific region and trying to collect EEG signal, you will end up with a data of size, number of channels, times number of samples into the size of the time window. So basically, this kind of data that you are collecting from the EEG signals is time series data. And this is a high dimensional data because you are working in this space. So for example, if you have 10 channels and you are sampling at 100 hertz, then you have 1000 samples per second, right? And if you're collecting it for 10 seconds, then you have that much number of samples. So it's a very high dimensional space to work. So that is one of the challenges that we face when we work in EEG data or in fact any other kind of biosignal data, primarily because they are digital sensors placed at various spatial locations. And when we are trying to collect this data in a digitized way at some sampling rate, you will have high dimensional data, right? So for example, if you are trying to capture data of the user planning forward movement for 10 seconds, right, you will end up with a very high dimensional matrix. And if you try to capture multiple such movements, it will be much larger. So that is one of the key challenges, that the data is high dimensional. So it is difficult to work with the data because you have to process the high dimensional data and you have to build a robust pipeline that is also low latency. So it is important to understand that the kind of systems that work with biosignals generally should have low latency because you should not have too much processing time in terms of the feature extraction and the machine learning pipeline you build. So that aspect also factors in as a result of the high dimensionality. Now, one other challenge that we talk about here is the challenge of spatial blurring. So what happens is, as I was mentioning initially, when we look at biosignals versus EEG, these are essentially derived due to firing off a population of neurons inside the brain, right? And you are capturing it from the source or from this scalp, which is a distance, a significant distance away from the source of the neurons, because it is not an invasive method. You are not going inside the dura. So the signals that are generated inside, by the time they travel to the scalp, there is blurring due to the spatial traversal that they have to do. And there is the dense dura matter between the skull that is outside the scalp and till the place where the signals have originated, that is from the firing neuron. And therefore there is blurring of the signal. So this is a challenge that you have to work with because you have chosen to work with EEG as a data form. The main benefit of using EEG is that it is convenient because it is not invasive and you do not need a medical procedure to get the implants done. But the challenge that comes with it is that there is blurring of the signals that you are getting, and therefore you need to build robust algorithms to be able to detect the intent. And as you can also imagine, that when you are collecting data from sensors, as is also common with many other kinds of physical sensors, there is common artifacts such as muscular artifacts or supply lines interference. So these kind of artifacts will also contaminate your signal when you are acquiring, because the user may not always be in a fixed position. He may make some other movements. There might be some other external noise factors that may contribute to affecting the signal quality. So therefore, all these aspects tend to build up and present themselves as challenges when we are working with EEG data. And one of the most important and in fact the most challenging aspects, these are all mostly physical aspects, but inherently by nature itself, these signals are non-stationary, low frequency signals. So when we look at non-stationary signals and these kind of signals that are low frequency, the challenge is what kind of methods do you use to process them. So there are various ways in literature that have been adopted for working with these kind of signals. And depending upon the computational feasibility and the accuracy of the system you want, you can choose these methods and accordingly build a pipeline. Now, in this presentation, we are going to cover a very specific set of methods, but you can generally always look at literature to understand what are the methods that have been adopted for various kinds of systems and how we can use these in the pipeline that we want. So I'll also try to explain the challenges in terms of how we could visualize them. So on this slide, what we are trying to show is that other than the physical challenges that we were talking about, we also have a lot of variability. So as I was mentioning in the previous slide, that there is non-stationarity in the data in these kinds of signals for EEG signals, EMG signals, and so on. One of the consequences of this is that there is very high variability. So if you look at data across subjects I mean across users. So for example, if I am one user and another person BEG you are collecting, there will be very high variability in the signals that you are collecting. Even across sessions. So what is a session? A session is a set of trials. So what is a trial? A trial is an action. So to give you an example, if you are trying to build a set of EEG signals for moving the wheelchair, as I was talking about, you have 4 sets of actions or 5 sets of actions, which are your forward, backward movement, left, right movement, and stop, right? So for each of these, you will first be collecting training data to train your classifier and build your pipeline initially. And you will conduct trials for various users. So these users are what we call as subjects. Now, trials are not always are not always done together. Right? Trials can be conducted in various sessions. So what we will do is, for example, we decide that the user will be able to move the forward direction for, let us say, 10 seconds in a specific trial. Then he will do backward movement for another 10 seconds and movement towards the left for 10 seconds and right for 10 seconds, right? So we may build a experiment like that for collecting data from users. And this experiment will be repeated multiple times. So each experiment is what we call a trial. And we conduct multiple sessions of those kinds of trials, right? So basically, since we want a lot of training data, we cannot always ask the user to do a very long experiment continuously trying to move. So we will conduct different trials in various sessions for various subjects. This is how we usually structure our experiments for collecting data for biosignal-based systems. So as you can understand that there is variability across users, that is something which we expect as well. But even for subjects or for the same user, you can have variability across multiple sessions and across multiple trials. So what I mean to say is that the signals for forward movement will not exactly be the same every time the user thinks about a forward movement, or the user's forward movement in one session may not do the same as the next session. So all of these variability aspects contribute to making it more challenging to building the signal processing and machine learning pipeline. So if you look at this figure, for example, this is a very common figure that shows the projections of the data. So what you can see in this figure is the kind of scalp map or your top view of your brain. And each of these dots in this plot, the black dots that you see, these are all recording electrodes. So it is similar to the 1020 system, but it is a very dense recording of a large number of electrodes. So you can also have up to 64 and 128 electrodes with high resolution system. And this data has been collected from those kind of machines or those kind of devices. And what we see here are the scalp maps. So this is essentially a plot of the power spectrum of the signal. So you apply some inverse localization.
Sir, your mic got turned off. You need to unmute yourself.
Yeah, you're able to hear me now, right?
Yes.
Okay.
Yes, sir.
I'll continue. So we were talking about challenges in working with EEG data, and I was trying to explain what these plots were. So when you are working with data, and these are the plots of the power spectrum or the band log of the squared values of the signal at the various spatial locations for different users. So what we are trying to do here is that one of the users is using the left hand for imagining movement of the left hand and imagining the movement of the right hand. And he is trying to do it multiple times. So each of these plot here is for one such trial. And these are aggregated for the sessions that are conducted for a specific user. So what we are trying to understand from this plot here is that there is a lot of variability in the signals. You can see that the activation or the region that gets the red color is what is of interest to us, because that is what gets activated. But you can see here that the region not only changes with various trials, but it is also different in terms of how strong the region is activated, right? So for example, this user, you can see that most of the region is blue, but for this user, there is a very prominent activation in this region. Similarly, so what is ideally expected here is that when you are trying to move the left hand, there will be a prominent activation in the right part of the cortex. In the right motor cortex, there will be activation. And when you're trying to move the right hand, there will be activation in the left motor cortex. So it is called as event-related desynchronization, where you expect that there will be very strong activations in the contralateral side of the movement that you are trying to imagine. But that if you look at actual data, you will see that it varies a lot. The activation region will vary significantly, and it will also not be the same for multiple trials or for multiple users. So as a result of this, it sometimes becomes difficult to really able to build a classifier and get the right set of features to build the machine learning pipeline. So one of the challenges here that you have to classify n events. So as I said, for the wheelchair, you have, for example, 5 classes, right? You may want to move forward, backward, left, or right. And for each of these events, you have to be able to detect them from the state of rest, right? So depending upon how complex the system you build, you will build more number of events or user intents that you want to detect, you will need to build a robust classification system to detect all n events. And as you increase the number of events that you want to detect, it will become more complex in terms of the classifier that you need to build. So for example, if the conventional way of 1 versus rest or 1 versus 1 is used, you can understand that 1 versus rest, for example, if you have 10 events, you have to build 10 classifiers. But if you have 1 versus 1, then you have to build 10 C2 classifiers. So depending on the kind of modality that you are adopting to classify these n different events or n different classes, you have to build that kind of a robust multi-class classifier. And one other common challenge is that there is bias towards majority class samples. This is basically to say that when you are working with biosignals, there is this problem of class imbalance that happens. So class imbalance is common because you are trying to detect multiple intents, and it is not very likely that the user will repeat all the events with the same frequency, right? So for example, if you are building a system to detect left and right movement, and you are also having to say detect the state when the user is not doing anything, right? So that state of rest also you need to classify. Now, for all these classes, the most common state will be the state of rest, because the user will not always be moving left or right. He'll usually only initiate movement when it is required. So you will have more samples of the class rest compared to the samples of the class left movement or right movement. And because of this imbalance in class, whatever classifiers you tend to build, they will have some bias towards the majority samples, where majority samples, I mean the set of classes which are large in number in your training data set. And accordingly, this bias also you have to mitigate when you try to build a processing pipeline. So that was primarily the challenges that we faced when we worked with EEG data, largely with respect to the initial challenges were with respect to physical limitations or nature of the data, while some of them are also related to the kind of classification or the kind of approach we design in building these systems, and accordingly we may increase or decrease the complexity of the system. So having taken a look at what are the challenges we face while working with EEG signals, we now try and see what are the different phases of an EEG signal processing pipeline in terms of what are the exact steps that are used. So in the initial slide, I had shown you a high-level block diagram where we try and say that these are the EEG signals captured from a device. We build some signal collection approach to digitize the signals. We have frequency-based filtering or some pre-processing approaches. We have feature extraction and we have classification. And in this slide, we are trying to understand what we do in each of these steps. So I said the EEG signal acquisition is a phase where you are collecting digital signals and you have a sampling rate for a time duration from n number of channels, right? So you have this time series data of various intents or actions that you want to detect from the user. Now you will take this data and you will run a pre-processing approach, typically to reduce noise or remove artifacts, and you will do some frequency filtering. The reason you do some frequency filtering is because depending on the type of activity that you want to detect, you may use a different frequency band. So, for example, eye movements and occipital activities are in a low frequency band of say 8 to 12 hertz. Motor movements, when you're trying to imagine left or right limb movement and so on, are in a relatively higher frequency than the occipital rhythm. So that is typically 12 to 30 hertz. And these different frequencies are called as different bands. So we have alpha band, beta, gamma, and delta. And where gamma and delta are the higher frequency bands, that is up to the range of 50 hertz. Or even, in fact, 70, 80 hertz are higher gamma rhythms. But more frequently, the region of interest in activities is largely in the lower frequency band. So we do some band pass frequency filtering in the pre-processing to identify the band of interest depending on what we want to detect. We use some artifact removal approaches. So in case we may want to remove supply lines interference, or we want to remove some constant noise that has crept into the signal. So accordingly, we also try to do some artifact removal. And epoch extraction is essentially the process of time windowing these signals. So as you are aware, in neural networks, when we train, we say an epoch is a complete training set of samples. In this case, what we are doing is we are generating a set of training samples by time windowing your data, and you are trying to identify the different segments in which you would like to generate a prediction. So that is where you are doing epoch extraction. Once you do this pre-processing, we will do the feature extraction. As I said, that EEG signals are high dimensional, therefore you need to build a robust pipeline to be able to extract features from these signals and build a classifier. So when we look at feature extraction, this usually has two components. One could be a suitable feature selection or a feature representation algorithm, and a dimensionality reduction or a feature extraction algorithm. So you will basically work with one of these, and or in fact even both in many cases, and try to build a lower dimensional representation of the data. Your initial data was in high dimensions, which is of the order of the number of samples times the number of channels and the time duration for which you have acquired the signals. And once you have taken the data, you would like to reduce it to some dimension which is lower so that you can easily build a classifier. This talk, we are largely restricting to classifiers that work initially in low dimensional spaces. So we are talking about kernel-based methods which will take low dimensional data and work with them in high dimensional spaces where there is separability. But for these kind of algorithms, we generally prefer low dimensional representations. More recently, we have deep networks that can also work with direct high dimensional representations, and that we will sort of briefly discuss in the concluding parts of the talk. But in this approach that I am presenting here, we are largely looking at conventional support vector machine kind of classifiers. So once you have done the feature extraction and dimensionality reduction, you will train a classifier. You will test its accuracy on a test set. So the offline training part is where you are training a model and building the model parameters or tuning the model parameters on a validation set. And once you have trained this classifier, you will use it for online intent prediction or on a stream data. So this is the training data part. And when you are using your classifier model in a live setting for prediction, it will get online EEG streams. You will time window it, do the same processing, and get the features for the test data and get predictions on the test data. And once you have predictions on this, you will use the predictions to actuate or do the actual intent that you want to do. So for example, if the classifier output is prediction to move forward, then you will move your wheelchair by a fixed range or fixed translation value that you have specified in your system. So according to the intent that is predicted by the classifier, you will identify what action or actuation has to be executed in the system. So this is broadly how the EEG signal processing pipeline will work for a BCI system. So I'll briefly review some approaches that have been there in literature for each of these kind of segments that I talked about in the previous slide. And here, this is largely to introduce the approach that we have proposed and try to put in some context about why we chose that approach. So these are from papers that are mentioned on this slide. One of the ways of doing this was that you take EEG data. As I mentioned, that it is of multiple frequency bands may be of interest, right? So you would do frequency filtering in each of these frequency bands. And then there is some methods for dimensionality reduction that we were talking about. One of these methods is common spatial patterns, that is referred as CSP on this slide. So what this will do is that it will take a high dimensional representation and convert it to a low dimensional representation. This is very similar to principal components analysis. Basically, you're trying to project it in the space of the eigenvectors. But one of the important differences between PCA and CSP is that in CSP you also have a whitening step. So that is required for these kind of signals. So there is a paper on CSP which you can always look up for details. But for the summary point of view, it largely is taking your high dimensional signals and converting it into a low dimensional space where you can do some feature selection. Now, it is not necessary that you might want to use all the feature selection, all the features that were generated by CSP. So you may do some feature set selection that has been proposed in this approach. Details are given in this paper. And then what they do is they use an ensemble of classifiers. So they have used SVMK, nearest neighbors, and various other classifier models. And they combine the output of these multiple classifiers as an ensemble and use it to get a prediction. So this was one approach in literature where we can see that they have taken multiple frequency bands, they have done dimensionality reduction and some feature selection, and used an ensemble of classifiers to the final prediction. What we can take from motivation from this approach is that you can look at multiple frequency bands, so you may need to look at different frequency resolutions for the data so that you are able to build robust pipelines. And secondly, there is an imbalance in the data that I was talking about. So one of the possible ways of addressing that is also by looking at an ensemble, so that if one of these classifier models is trained well, that will probably give you a prediction with higher confidence. And therefore, when you take it from a variety of multiple classifiers involved in the pipeline, the general performance may be better. So they have used some approach for A, handling multiple frequency scales, and B, trying to look at class imbalance. Another approach that has sort of been done here, which is a related kind of approach only in terms of the initial steps, that they are also doing frequency filtering. They have done common spatial patterns for dimensionality reduction. And after that, they have used the same kind of classifier, but they are fusing the score from multiple frequency bands using a combination approach. And from the fused score, they are computing what is the actuation to be done. So this, except for the machine learning or the classifier part, the rest of the pipeline in both of these cases is same. The motivation from both of these methods is largely that you would need to look at multiple in the presence of data imbalance. So I hope by now we are clear that when we work with EEG data, we need to look at different feature scales, we need to look at different frequency bands and low dimensional features in each of these frequency bands for our purpose of interest. And once there are different bands, we also need to employ some selection method, right? So either we can run all of them to different classifiers and try to build this score, or what we can do is we can try to do some kind of selection here itself from the multiple CSP bands, so that we have a good set of features, and then we train one good classifier that does robust classification even if it doesn't class imbalance. So this is what we try to do in our approach, where we say that till this part, it is similar, that you can look at frequency representations on multiple scales from the data. And now you try to build classifiers. Before doing the classification part, we employ a simpler feature selection approach here, so that we have a best set of features from the CSP. And then you use a twin support vector machine as the classifier, which we will discuss in the coming slides. So with this, we propose, or I discuss the approach that was proposed in the paper, where, as I was mentioning so far, we have common spatial patterns for feature extraction, which is very common. We have not changed anything additional from this approach that is already well documented in literature. Now, once we have features from CSP on multiple frequency bands, we have the CSP features. Now we have to combine or do some selection of these features, for which we have used a measure called as classifiability, which is a simple selection measure to compute which are the robust set of features that we have obtained. So once we have the best set of features, or essentially we select which band has been the best, we will then use what is called as a twin support vector machine, which is robust to class imbalance and therefore will result in better classification accuracy even when there is imbalance. So this was a motivation based on what is available in literature. We wanted to see if we can use a robust or a simple method for selecting the features, and then a robust classifier which is imbalanced, which is not affected by imbalanced data, or which performs well even in the presence of imbalance. So that is where we try to use a twin SVM, and we also show that it has lower computational time. So our entire pipeline, starting from multiple frequency bands to common spatial patterns, and then computing the final classification, is also shown to have lower computational time compared to the approaches that we were seeing on the previous slide. So we'll talk about numbers in the slides, but I would just like to explain what are the motivations that have been there. So as I said, EE signals have been pre-processed in the band of 8 to 30 Hertz. So we are looking at data for movement, right? Left and right movements or imagined movements. So that region corresponds to what is called as the motor cortex, and it is in the 8 to 30 Hertz frequency band. Now, instead of looking at this whole band together, what we did is we built smaller frequency bands. So for example, we took 4 Hertz bands in between this. So 8 to 12, 12 to 16, 16 to 20, and so on. So we constructed subbands. We filtered the data in each of these individual subbands and applied common spatial pattern filter to extract discriminative features in each of these subbands, right? The next task, what we want to do is to find out which is the best frequency band or the optimal frequency band that has given the most robust CSP features. So one way is to do feature selection using conventional approaches, where you try to evaluate a model with selecting these features iteratively, and you try and see which is the best set of features that has resulted in good classification performance. But alternatively, what we propose here is to use classifiability, which is a simple method to compute which set of features determines the best separability for the classes without actually using a classifier in the pipeline. So ours is a distance-based metric, which is called as classifiability, that has been used in other contexts for decision trees in a related paper, but this is the first time that we use it for feature selection in this pipeline that we're talking about. So why we should use classifiability? So let us say this is a synthetic data set in 2 dimensions, right? You have these 2 dimensions, and this data belongs to 2 sets of samples, 2 sets of classes. One set of classes is denoted by blue, and the other set of classes is denoted by pink on this plot. And if we were to visualize this data in a 3-dimensional space that is shown here, where the 3rd dimension is the label, right? So what we have done is all the points that have class 0, for example, that are the blue dots, those we have represented by 0 on the 3rd axis, and whatever class or samples that correspond to class 1 are shown in this plot, the pink samples, that we represent as 1 on the z-axis. So if you look at this plot in 3 dimensions, where the initial 2 dimensions are the dimensions of the data, and the 3rd dimension is the label, you will see that there is a good separation in the data if the classes are well separated in this space. Right? But if these classes were not well separated, so here you can see that the pink and blue points are all over the place. You really cannot distinguish where is the exact region for the class 0 or class 1. So in this case, if you try to plot a 3-dimensional plot of the samples and the label as the 3rd dimension, you will find that this surface is very fluctuating. It is not varying. There is no smoothness in this surface. And in both cases, we will come to how we compute classifiability. But in a separable data set, our metric for classifiability is very high. It is close to 1. And for a non-separable data set, the classifiability will be low. This is how we want to use this method. So if we have these common spatial pattern feature representations, we want to use classifiability as a method to figure out how separable the data is. Accordingly, the value of classifiability will be high, and we will choose that frequency band for which the classifiability value is high. That is the motivation for using classifiability. Now we'll come to how we compute this, and this is on this slide. You can see on the right here that we have samples of 2 classes, right? We have samples that are of one class that are represented by cross, and samples of the other class that are represented by circles. And there is a parameter in classifiability that you choose, which is the radius of interest. Okay, and for each sample or each point in this data set, you compute a co-occurrence matrix for each data point. The co-occurrence matrix is a 2 by 2 matrix, where each element of that matrix wjk is a distance of a pair of points in the data set, right? So for each point xi, we will compute this distance metric for all other points, sum it up, and then use an integrator function and set the value as 1 in the matrix if it is less than that threshold or 0 otherwise. And d is a neighborhood distance, which is the parameter that is chosen by the user. So basically you can see here what will happen is if you are trying to find that there are too many points which are close to a specific point, the indicator function will tend to go to 1, and accordingly you will have high value of this element in that matrix, right? So for that particular sample, the value of wjk will be high. And so we compute this co-occurrence matrix for each point. Basically, the 4 values that you see here will be the number of points that are closer for the same class and for the other class, right? So number of patterns of class 1 with radius of pattern P1 is 4. That is the first element. The second element will be the number of patterns of class -1 which are within the radius of that sample, which is 2. And the number of patterns of class 1 which are of radius d with the other set of classes samples, that will be 1, and for -1 it will be 1. So we have to count how many samples of that class are there around this or inside this region for the same class and for the other class. And similarly for the other set of samples for the same class and the other class, and we will construct this co-occurrence matrix this way for each sample and add them up. So once we have added the co-occurrence matrices for all the samples, we will normalize them by dividing it by the number of samples, and we will compute classifiability as the sum of the diagonal elements minus the sum of the off-diagonal elements. So basically it is kind of a trace value minus the off-diagonal elements, and this is what is the classifiability value that we were talking about in the previous slide. So if you will see that there is good separability in this case, if you compute the overall classifiability for this kind of a data set, the values will be close to 1 because the diagonal values will be higher and the off-diagonal values will be relatively lower because samples of the other class are far away from the samples of the class of interest. So in this kind of scenario where the data is well separated, the classifiability will give you a higher value. But if it is not very well separated, you will have more confusion in this region. Therefore, the off-diagonal values will also contribute to the computation of classifiability, and the eventual value of C will be lower, right? So this is how we can use classifiability as a measure to compute how good a particular data set is. So I'll briefly pause at this point if there are any questions. In understanding how this is being calculated, you're welcome. And otherwise I'll move on to how we have used this afterwards. So we are good so far? So this slide sort of illustrates how the classifiability changes with respect to the distance of the sample. So this is a synthetic data set that is very well separated, right? As I mentioned here that there is one parameter D that you have to choose. D will determine how large this circle is, and if you are looking at a larger area and there is more confusion, your classifiability value will be poor. So the value D has to be well chosen. Now we illustrate how the value of D affects the kind of data set in these two slides. So if you have a well separated class distribution, so all sets of say class 1 are very well separated from samples of class -1 in this plot. And here if you see for different values of the neighborhood distance, this is how the classifiability will vary. So you will see that for lower values, the classifiability will be very clearly 1. And this, as you sort of increase classifiability, the region of confusion will grow. Or in fact, as you increase the neighborhood distance, the classifiability value will change and the region of confidence may drop. But for a well separated data set, for low values of neighborhood distance, you will be clearly be able to see the classifiability as a useful parameter for trying to understand how separable the data is. And if it is not very well separated, if you can see in this case, the samples are all over the place and you try to compute classifiability, even for low values of neighborhood distance or high values of neighborhood distance, the value will not change much, right? So in such cases, you will, whatever value of neighborhood distance you may choose, the classifiability will not be very good. So if you can average it over multiple values and see if it doesn't change much, that means your data set is not good or your feature space representation that you are using is not very good, and therefore you should choose a better value, better or a different set of features, because the classifiability in general is low, right? So I hope these two slides have made it clear that if you have a well separated data set for low values of neighborhood distance, you should get high value of classifiability, therefore you can separate it. And if you have very distributed space where the features or the samples lie, then the classifiability, irrespective of neighborhood distance, will tend to vary around the same set of lower values. So it is not, in general, if you will see, it is not a very well separable data set, irrespective of whatever neighborhood distance we choose. So once we have used classifiability, we have selected what are the set of features, right? So at this point, we are here in the pipeline. We have taken the set of we have chosen the best set of classifiable features from the EEG data. And now we want to train a model to do the predictions. So as I was talking about the fact that we usually use support vector machines, how support vector machines will work is that you have this representation in these two classes, as you can see here. And here you can see that there are more samples in the red class, right? This is a common situation for imbalance, where you can have more samples of one class compared to fewer samples in another class. You can see there are more red points here, right? So this may correspond to a real situation where in a BCI system, these are the points where the user is trying to imagine forward movement, and these are the points where the user is not trying to imagine anything, or if they're all the other points, for example, right? So obviously all the other points will be much larger than the smaller number of points of the class of interest. So when you're building a one versus rest classifier using an S3M, what will happen is that the plane or the separating plane will tend to be biased. It will tend to move towards the samples of majority class, right? And once it is closer to samples of majority class, what will happen is that when you're trying to use this kind of a model to get predictions, you may tend to invariably have a bias towards predictions of the majority class, and therefore your classifier will not be very robust. So what the SVM basically tries to do, as many of you would already be familiar, is that it tries to solve a maximum margin hyperplane by finding a weight vector which has the minimized L2 norm here, and you have slack variables which allow you a trade-off between the support vectors being at unit distance by a parameter epsilon, which is weighed by a user supplied parameter C. So epsilon are the slack variables, and the constraint to this is essentially that the product of the distance of the sample from the hyperplane multiplied by its class level should be greater than 1, right? And if you are using a soft margin version, you can allow tolerances according to the slack variable, and all these are positively constrained slack variables. This is the common formulation for SVM, and the separating hyperplane tends to be biased towards samples of the majority class if there is very high bias in the data set. So in our approach, where we want to use a one versus rest approach for classifying these biosignals, it is sort of difficult to use the SVM. What we are alternatively proposing is a paper which appeared in IEEE Transactions of Neural Networks and Learning Systems way back in, I think, 2010, that is using a twin support vector machine. So what the twin support vector machine does is that instead of finding this one plane which is a maximum margin hyperplane between these two sets of samples, the twin support vector machine is essentially trying to find two non-parallel hyperplanes. So you can see that there is one plane which is close to samples of one class, and another plane which is close to samples of the other class. And instead of using that one maximum margin hyperplane, we have two non-parallel hyperplanes. And the way to find this is that the formulation or the optimization problem changes here, right? What is the motivation? We say that this plane, the blue plane, should be as close as to samples of the class indicated in blue, and should be at least unit distance from samples of the other class, right? So in the typical SVM, you say that the separating plane must be at least unit distance from the support vectors, right? So here what we are saying that instead of the entire data set, we say that it should be at least unit distance away from samples of the other class. And similarly, this red plane will be as close as possible to samples of the red class and at least unit distance away from samples of the blue class. So this is how these problems have been formulated. Here you can see A and B are the samples of the two classes, right? Capital A denotes the set of blue samples, for example, and B denotes the set of red samples. And E is a vector of months. So you are trying to minimize the distance of the plane. So this, for example, will be W1
x plus b, or this is the weights and bias of the blue plane, and W2 and B2 are the weights and biases of the red plane, right? So you are trying to minimize the distance or the L2 norm of the plane with samples of that class. So this is to say that it will be closer to samples of the blue class, and it should be at least unit distance away from samples of the other class, right? This plane W1x plus B1 should be distant of, it should be at a distance of at least 1 from samples of the other class, where this is the B class. Similarly, the other plane, that is this red plane, should be as close as possible to samples of class B. Okay, W2x plus B2 should be closer to samples of B, and it should be unit distance away from samples of class A. So if you solve this kind of two problems, you will get two planes now in the twin SVM, which are W1x plus B and B1 and W2x plus B2 that are shown here. These are the model coefficients. And you can use each of these planes to compute the test prediction. How will you compute a test prediction? You will have a test sample, you will see its distance from this plane, and you will compute its distance from this plane. And you will see whichever is closer, that is the class that you will assign it to. Okay, this is how the twin SVM works. So basically, of course, you can also do a dual derivation that is there similar to the SVM, but I'm not going to walk into details of the derivation here, because that can always be looked up from the papers, either on the twin SVM paper or on many other papers that have used the twin SVM. But we can also construct a Lagrangian and do the KKT conditions to get a closed form dual solution that is easier to solve, similar to the case of the SVM. So what we have in the twin SVM formulation, if we choose a kernel matrix, and the dual that we solve from the primal formulation is shown here. So basically you can have a much more simpler formulation using the dual, and that also allows you to use the kernel matrix similar to the case of SVMs, right? And this is a dual from the primal that you get, and this is a much more simpler formulation compared to the constraint and objective in the primal form. So the advantage of using the dual are the same as that is also there in SVM. You can use kernel matrices, and the computation is much more less in terms of the constraint. It is simply a bound constraint, and your objective function is more closely represented using the kernel matrix as well. So this is how you can actually use the twin SVM to solve for two smaller hyperplane problems. So you can see here that in this case, you have the whole data set to be solved in the constraints. But in this case, the constraints have samples of only one class, right? It is not the entire data set. Similarly, the objective function has to be solved only for samples of one class. So in that sense, it is a slightly smaller problem compared to the conventional SVM. And you are solving each of these two problems, but each of these are smaller subproblems. And that is why the twin SVM is beneficial for this kind of settings where you have data imbalance, right? So we walked through how the twin SVM works. I hope by now it will be clear that now we have two non-parallel hyperplanes, which is why the twin SVM is better for generalization. It is suitable for multi-class scenarios because, right, you do not have that bias towards a specific set of samples of a particular class, and therefore you can use it for multiple number of classes with larger number of samples compared to the conventional SVM. And you can solve two smaller sized QPPs with the twin SVM, which makes it faster as well. So compared to the SVM, since you are solving smaller problems, although there are two problems, but still it is faster in terms of each of the size of these smaller quadratic programming problems that you are trying to solve. So this was what the twin SVM resulted in an advantage compared to the conventional SVM. Now, here is to summarize what we did. This is basically the entire approach that I was talking about, from starting from the signals, using classifiability as a measure to compute the separability of the data, and then using twin SVM as a classifier that is robust to samples, imbalanced samples in the training data set, right? So you have this data, or you have, these are actually the feature representations that I was talking about, where you have used common spatial patterns or a suitable dimensionality reduction technique after principal components analysis, or CSP in this case. And then from each of the CSP features of different frequency bands, you try to select which is the best frequency band, right? So to see the label and evaluate what is the best frequency band in terms of classifiability, and according to choosing the this corresponding set of features which has the highest classifiability value, you can then train a twin SVM on that data and the twin SVM will obviously have two hyperplanes, and you can compute distance of the sample for the twin SVM, and whichever class it is nearest to, that hyperplane distance value will be the minimum. So that is how you can obtain the prediction on the test sample, and therefore result in high accuracies in predicting multiple actions even with noisy data. By noisy, I mean the imbalance due to the high number of samples in one class compared to the other, right? So this is broadly how the approach that has been proposed will work. You will have CSP features after frequency filtering. You will compute classifiability to determine which set of features are the best, and you can use twin SVM for the classification in the presence of imbalance, and it will be a robust classifier even in the 1 versus rest or 1 versus 1 setting also. But preferably you will use 1 versus rest because you have only those many number of classifiers as the number of samples, right? You do not need to build a combinatorial number of classifiers. So in such cases, this is how the twin SVM will help you in trying to find more robust classifier models. So to look at some results of how this approach works, we have compared it with the set of approaches. So this data set that we have used is the BCI competition data set that is available online. You can very clearly search. There were 4 competitions, and each of these had multiple tasks. We have looked at tasks that are related to motor imagery, because I was talking about movement, about left and right rims, and how we'll be able to build a pipeline for doing that. And we compared these three approaches, so filter bank, CSP, bispectrum, and DDFBS, that were shown on the previous slides where I was comparing it with different approaches. And we have also, so these are mostly in terms of what are the features extraction approaches, right? They have used filter bank, CSP, or they have used bispectrum or DDFBS kind of frequency data extraction algorithms. And even in terms of classification, we compare our approach of using the twin SVM with the standard SVM and recurrent quantum neural networks. So this, in terms of paradigms, we can see 2 class and 4 class. So 2 class was left and right imagined movement, and 4 class was left, right, and then foot and tongue, 4 set of movements that were imagined by the user, and the data set for the EEG of those users was generated. So this is a summary slide. Basically, we are just trying to show how much was the percentage improvement using our approach compared to what was existing. And you can see that in some of the cases, the improvement was high as 43% and 34% in some cases. And generally, we did perform well. And to look at specific results for these subjects, so there are 2 data sets that are shown on this slide, right? One of them is the BCI competition 4 dataset and the BCI competition 3 dataset. So if you look at specific results on the BCI competition 4 dataset, this has 9 subjects, and for each subject there are 3 sessions of recordings, right? And in these sections we take different frequency bands of interest. So we have done frequency band separation for all the 4 hertz bands in the 8 to 30 hertz band, right? And this shows which was the best frequency band selected for that user. So the basic idea of our approach, also what it shows here, is that you can also select which is the best frequency band that is suitable for a user, and therefore address the kind of variability issues that you face when you are working with multiple users. So this kind of frequency filtering in different frequency bands and then computing classifiability of the features in the respective frequency band will improve the model that is built for a specific user, and also address the problem of variability that you face. So this is what we got in terms of classification accuracies for our approach. And there is a computation metric called kappa value for these kind of systems that is reported on this data set. The computation of kappa value, you can refer to any literature. It is basically higher if the precision on that set specific intent is higher, right? So that kappa value has been shown on this for various approaches that were discussed in literature. And on the average, if you see, although for some slightly not beating all the approaches, the average value in case of, so this is the best frequency band, right? This is not a specific approach. Like this is only if you choose the best session and try to compute the kappa value. But all the other cases, this value is fairly high compared to the other approaches. But in terms of individual users, if you will see, we have higher performance for more cases than other approaches. This is because we are allowing to use an approach for classifiability that will allow you to select the best set of features for a particular user, and therefore you are tuning and making the model more accustomed to that user's frequency band, which is very important in these kind of systems, so that you can have good performance for the individual users, right? So, for example, in the best column, you will see that there is very high value for some of these, which is what results in the high average values. But in terms of consistency or more number of users where this kind of approach works well, the kappa values are higher for that case. Right, so this kind of approach is better to address the variability that I was talking about, where you have to tune the model or tune your processing pipeline so that you can have good, robust models for individual users. So that is the case where our approach comes in to play a better performance compared to the other state of the art. And in fact, I think even on multi-class data sets that we were talking about, where we have 4 intents that we were talking about in the previous slide. So these are the 4 intents I said: left, right, foot and tongue, and versus rest. This is the state of rest that you want to baseline against. There are 2 subjects for which the result is shown here. The frequency band that has been chosen for these users is indicated here, and this is the leaf SVM and this is the twin SVM. The rest of the pipeline is same. We have used common spatial patterns for feature generation, and we have selected the features using classifiability. In fact, selected the best feature in the best feature set in the corresponding frequency band, right? That basically allows you to choose the best set of features for the user and then use a twin SVM to build one versus the rest classifiers for that user. And therefore that has good performance compared to what we used to get using a standard lib SVM classifier, which is a normal L2 norm SVM implementation that is available publicly as well. Even in terms of the other subject, you can see that the twin SVM performs better compared to the standard lib SVM in a one versus rest setting. And the suitable frequency band is, of course, different for the different users, which you can see here, which is well identified and reflected in the results as well for our approach. So this was a walkthrough of the approach for frequency filtering based classifiability for use in feature selection and the twin SVM for classification. And as I mentioned before, that you will obviously wonder whether this results in more computational time compared to the conventional approach. So we took this kind of an approach that I was talking about in the beginning, where they have different CSP features for each of these values. Classifiability, I mean, we do classifiability selection and they do feature selection using a model itself in this pipeline, and then they use an ensemble of classifiers. So this is a place where the entire pipeline for a specific user took around 91 seconds. And in our pipeline for that same user, where we are using classifiability and training the twin SVM, we sort of completed in 37 seconds. So even in terms of the processing pipeline, we can see that our approach is, in terms of computational time, lower than the baseline approach that was used in literature. So this kind of approach not only results in good generalization or good model performance for individual users, but even in terms of computational time, it is tractable. What we are trying to show here are the 2 cases that took the maximum time for our approach. So one was the feature selection and the classifiability. These do not take too much time, as it is indicated the most highest this list is sorted in descending order, the time taken for each of these functions to execute, right? So the overall time is also lower, and that sort of makes it viable for implementation where you have low latency as a requirement. This is the entire pipeline in terms of computation time for the pipeline that we have proposed. So this was the first part of the talk, where we tried to see the EEG signals for different kinds of motor movement, and we tried to build a pipeline using classifiability and twin support vector machines for EEG data for the context of motor imagery based detection. So at this point, I'll sort of take a brief pause if anybody has questions. The next part of the talk will be on electromyograms or surface electromyogram signals for prosthesis, and we will look at a similar pipeline for that as well. So any questions, if there are there, I'll take a brief pause for that.
Sir, there is a question in chat box.
Okay, let me try and get that. Okay, so support vector machine is a classifier. Basically, if we talk about various kinds of classification models, right? You have come across neural networks, you have come across different kinds of supervised and unsupervised classification algorithms. So the basic area of machine learning, when we look at it, we say that you have different kinds of problems, right? Some of these problems is where you would want to define, identify what kind of a task it is, right? So if I am moving my left and right hand, and you want to find out whether I'm moving my left hand or right hand, that is a classification problem, right? You have to say whether it was my left hand or right hand. Therefore, for example, if my left hand is 1 and right is 0, you can predict 0 or 1. So you have a model that will predict the classifier or the kind of action that you perform. So this is a classification problem. On the other hand, if you may want to predict a real valued function. So for example, if you want to predict what is a value or the price of a house in a specific area, right? So you will have data for various houses. You will have their area, the number of rooms, the kind of locality, the per capita income, and so these set of values for different houses. And you will have one value of interest that you want to predict, which may be the price of the house. So the price of the house can be a real number. You can have the price to be, let's say 5 lakhs, or it could be 20 lakhs, depending upon the price of the house. So compared to what I was saying before, where we have a classifier, this is a regression setting. Okay. Now, in both of these cases, we use different models to build the prediction, right? You have to have a model which will take this data, that is, it will take the data of the features of the, for example, if you're talking about the price prediction problem, then your features are the number of rooms and the per capita income, the number of, let us say, rent payers in the house, and so on. So all this data, you will feed it into a model, and you will get the price of the house. If you are looking at a hand movement problem that I was talking about in the previous slides, you will build a classifier for the data, the kind of EEG data. You will take it into a specific computational unit, and you will output whether it is a left or right hand movement, right? So support vector machine is one such classifier. It can also be used as a regressor. And what we are trying to see here is that if you can see this slide where I was trying to explain what an SVM is, these are the set of points where, let me just go back to that slide 1 second. Classifiability, yeah. So these are some set of points for the left movement, and these are some set of points for the right movement. And you want to find out this line, okay? So this line, in this case, it is very simple because it is two-dimensional data. This is a line. But if you have high-dimensional data, if you have more than 3, it is difficult to visualize, right? This can be a plane. This will become a hyperplane in higher dimensions. So support vector machine is an algorithm which solves this optimization problem for this data, and it tries to predict which class you have, right? So it will basically find the equation of this line in this case. But if you have higher dimensional space, it will find the equation of this hyperplane, okay? And then if you have a new test sample, you will be able to see which side of the plane it is on, and therefore you can say whether it is blue or it is red, or in terms of real actions, it is right hand movement or left hand movement. So this model that we obtain, this equation of the line, is what is essentially one way of getting that line is through a support vector machine formulation, which solves this problem. And why it is called a support vector machine is basically because you will, as I told you, that this line, the condition is that it has to be at least some distance away from the set of points, right? There's a unit distance away from support vectors. And that is why this line, it is called as a maximum margin plane, which is at least some distance away from samples of both classes. Because if it overlaps, then it is not a very good separator, right? And these points that are close to these are called support vectors. So you need to find the set of points that are closer to this separating plane, and that is why it is called as a support vector machine. Vector basically because all these points are multidimensional. So in some dimensional space, so these are all vectors, and you need to find support vectors and a separating plane in between them. Okay, so that equation of the plane is your model that you have. So similarly, in this case, the problem that I was trying to show is that you have smaller classes here and larger number of points here. So this line, if you try to find out, it will be closer to this. Okay, so every time you try to find the distance of a test point, it will be more biased towards this. But you want that if it is of this point, it should be detected accurately, right? So that is where we say that the twin support vector machine is a better alternative, where we have two lines or two hyperplanes that we find, and this is how the formulation is. Okay. More details, of course, you can always read the paper to find and understand, but this is one classical algorithm of how we have approached this problem of classifying in the presence of imbalance, right? And this is what I was trying to show. I hope I have been able to answer that question. I have basically just tried to provide details on a higher level, but of course there are always more intricacies with respect to the formulation and how it is solved for a data set. All those you can always look at the paper and try and understand what works the best. Okay. So any other question at this point? All right. I'll move forward. So we have looked at one way of building a system for developing EEG signals. Okay, brain signals you classified, you conduct feature extraction and build a classifier, and finally control the device using that. So this approach was basically what machine learning approach you can use to do that. Okay. Now this next part is trying to look at surface electromyogram classification using twin support vector machine. So this problem is similar, but the data is different. So here we are using surface electromyogram. As I said, that in various patients you might have seen, or various users or people, their limbs may have deteriorated in function and they may need to have been amputated. So they do not have their hands or feet, limbs that have been removed due to various reasons. And but the activity of the nerves that is there around that region, that is still available. So therefore, signals can be collected from those areas, and that is what is called a surface electromyogram. And then if you can build this kind of pipeline using the approach that I was talking about earlier, and you can build a robust classification pipeline, then you can use it to control these prosthetic arms. You have seen these robotic arms that are available, which have multiple movements that you can support. And if you can detect what movement the user is trying to think about from the nerve activity, that is this kind of system based on surface electromyogram. So how this works is, so as I said, for some user, this hand may not be there, right? This is just a representation. But if the limb is not there, there may be connecting nerves that are there, right? For example, these muscles, so nerve you cannot directly connect to, you always read it from a muscle or muscle actuations. So for example, there is a system from Touch Bionics, there are various other systems that have sensors you can place here, and look at 4 muscles in this study that we had done. We looked at flexor pollicis, flexor digitorum, flexor carpi radial, and extensor carpi radial and ulnaris muscle. So these 4 muscle movements we tried to capture for users and build a system so that even if they do not have their physical hand, they'll be able to use a prosthetic arm that will be worn around this place, and then that will sort of help them realize the functions of the arm. And what all did we try to realize? We tried to realize 16 classes of movement. Okay, so you have that kind of prosthetic arm here, right? And you want to see what all ways you can move this. Okay, so we took these 16 different classes. 0 was rest. So whether you want to move the little finger, ring finger, middle, or index finger, thumb movement. Okay, little and so this was all single finger movements. Then you have pair movements. Okay, little and ring finger, ring and middle, little and index. So these kind of movements. Then there was wrist flexion. Okay, wrist extension, and combined wrist flexion and extension was one class. Moving all fingers for an action, for example, a clasp kind of action, a grasp kind of action, and a pinch grasp kind of action. So these were the different kind of movements that we tried to identify from data that was collected from these locations. This is what is the idea of what we are trying to do here. So we want to build an algorithm to control this kind of an arm from data that has been collected at these locations, and our algorithm will essentially classify it into one of these states, these 16 states, and correspondingly there will be a movement of this limb. So the user can actually control the device from muscular movement or by thought. So this study was done in collaboration with the Royal Melbourne Institute of Technology and IIT Delhi. And the work that the data set that we collected, we had 9 users. 8 of these were healthy users because we initially did not have too many amputee volunteers. One was an amputee volunteer who had amputation on the right forearm. Okay, and then there were 6 males, 2 females. These are the average age and height of the subjects. And this generally they were all healthy subjects, except for there were no significant changes in terms of their laboratory or vital measurements. So we recorded the data for 50 seconds. Initial 5 seconds was an adaptation period. Then they perform that action. So when they perform this action, I mean to say that they will perform one of these movements. Okay, and this will be for a duration of 5 seconds. And then they will rest after that, and then again 2.5 seconds they will start, they will do it for 5 seconds and rest after that. So like this, they conducted multiple trials of 10 seconds, and during each task execution, the subject repeatedly performed the action for 5 seconds. Okay, so this is how we collected that data, and this data for 2 sessions per subject per task was collected. So we had 40 trials per task. So we had these kind of signals. So there are 4 locations that are shown here. Right, and for each of these 4 locations, you have a digital sensor that is collecting the signals. So this is how you have these 4 channel-based data, and it is coming over time, right? So you want each of this is actually what the user is doing. So for example, if the user is trying to do little finger movement, okay, so in this plot, every time the trial is showing is doing the movement of this little finger, okay, and we are collecting all this data. So this kind of time series data we have collected it here. And we will do pre-processing, right? We'll sort of remove some artifacts or noise that is available in the data, and we will do a pre-processing on the data. We will perform feature extraction. So in this case, we have not used a very complicated system of feature extraction, although that can be done. But in this specific study, we use very simple RMS values as features or root mean square values. And then for classification, as I mentioned, we are using the twin support vector machine, because obviously even in this case, you have to identify these 16 categories of movement, right? So therefore, there will be a lot of different classes, and there will be a lot of imbalance in the data. That is why we are trying to use the twin SVM for classification. So we took this data, we built our model, we trained it, and then we tried to get predictions on the data. So for these subjects that we had, this was the kind of accuracy that we got for twin SVM compared to libSVM, that is the standard L1 norm SVM. So you can see that for the twin SVM, for these various kinds of movements, the accuracy is relatively higher compared to the libSVM performance or the standard SVM performance. And even for each subject, if we average the performance across multiple tasks, the average performances are also better for the twin SVM compared to the standard libSVM. So in this case also, we have shown that this twin SVM is better for this kind of approach, and we have been able to build a system for moving a prosthetic limb from a user using the data that is collected from the muscles in this region, and the user can actually do one of these 16 movements with this kind of accuracy. This is what we want to show in this study, and of course we are also able to use it for some patients, and they were able to improve their physical activities of their hands by using this kind of an approach. So this generally helps them for rehabilitation, and there is a lot of training involved. It is not that in the first go itself it will be very easy. You have to collect training data. Once you train the model, then we ask them to use this system, and therefore at that time they are able to perform well. Okay, so that is what we were able to show in twin SVM for EEG and other SEMG data. So for EEG data for BCI systems, we tried to show the performance, and for surface electromyogram data also, we tried to show how this is important. And of course, there is a lot of papers and work on twin SVM variants that have been proposed. Now, towards the last 10-15 minutes of the talk, I would like to talk about what other things we can do, or what are the future directions for this kind of work. So we will look at 2 aspects. One is how we can use neural networks based on the twin SVM kind of idea. And secondly, we can also have sparse models based on linear complexity machines. So I mostly generally talk about this twin kind of principle for neural networks and how we can extend that to neural networks. At this point, so rest of the talk, there are only 2 more slides about future work. So any more questions that you may have, I'll take at this point, and then we'll discuss what are the future directions for these kind of projects and these kind of experiments. So if you are good enough, I'll continue. Now, in terms of, so what I showed you with the twin support vector machine was basically a classifier with a support vector machine. It was basically a kernel kind of formulation where you are taking the data in low dimension and you are trying to map it to a kernel space, and you are trying to determine separability as a result of the kernel projections of data. Now, when we look at newer methods of machine learning, there are a lot of deep neural networks that are emerging, and therefore you can sort of extend this idea to deep networks also, or in fact to neural networks also. So as you can see in this figure, we are trying to show it for a simple case where there are two classes of interest. And instead of now using one network, we are saying that we have two networks. Each network will try to optimize the projections towards samples of one class, right? In the objective function for this network, we will try to get it closer to samples of class A. And in the function or the objective function for this network, we'll try to get it closer to samples of class B. And in the final layer that we have, we can aggregate the predictions as similar to what has been done for a twin support vector machine. But in this case, these coefficients that are available here are the projections from the penultimate layer of the network. So the pre-final layer, we'll take that as a projection mapping, and then we'll compute a linear separator in this space. Similarly, in this space, and for a test sample, we will see whether it is closer to this or closer to this network, or actually, in fact, this distance, and therefore we can assign it a sample of class A or class B. So this is how a twin network architecture can be realized, which is motivated by the approach that we had followed for the twin SVM. And this, we have shown that there are a few papers that have used this approach for EEG data, and it has shown to be better also. That is the reference that is given here. So the same kind of approach that we were talking about earlier, that you can do features if you want to extract CSP features, that is one way of doing it. But if you want to directly work in high dimensional spaces with deep networks, that is also possible. And therefore you can use this set of multiple network models in a twin kind of sense to aggregate these predictions and build robust models. So all the current research that is coming up in this area is focusing on using deep networks and those kind of architectures. We will see that it is more feasible in such situations where there is imbalance in data, if we can use these kind of twin models for building the predictions and building the systems. So that is one of the directions using twin network models that we can talk about. And as of course, you can also understand from this that the complexity and the training time will increase for these kind of models. But of course, these are also suitable for larger data sets because you do an epoch kind of training with different batches. And therefore, mini batch kind of approaches can be employed for training each of these individual networks. This was one direction in which work is going, where we want to use larger and deeper models for biosignals so that we can cater it to a large number of users and build more subject independent systems. So instead of having a model for each user, whether we can have models that work well for multiple users as well, and whether those can be realized by these kind of twin architectures and deep networks going forward. So this is one direction in terms of machine learning approaches where we can improve and we can build more robust pipelines for this biosignal processing. And one other direction that I would like to talk about is development of standards. So this field of brain-computer interfaces or brain-machine interfaces in general is a fairly recent area in terms of how work has evolved and is now moving towards industrialization. Mostly it was research work that was confined to labs and research labs, but now of course it has started to move into commercial products and even in catering to industries as well. So therefore there is a need for requirements in terms of standardization. So for example, the kind of data that you collect from the brain from multiple devices, whether they should have standards for interoperability, or whether you collect data from one device, it should adhere to a specific format so that the data can also be read and processed by another device or another processing platform. So these kind of challenges that we look at in terms of BCI or brain-machine interfaces, these are addressed by developing standards. So there are various bodies that are working on this. One of the examples is that the IEEE itself is involved in developing standards, and there are working groups that are currently working towards building standards. And these are some of the aspects that they talk about. So on this slide, there is a link to the IEEE Brain newsletter, which sort of shows and a primer on what the standardization objectives are and what we want to do with building standards for this, right? So there are various aspects of this whole pipeline. And there are 6 different aspects that have been identified by this working group. One of these is called sensing technologies, where we want to see what kind of data we are collecting, what is the modality. As I told you in the initial slide, there is a trade-off between space and time. And so what kind of sensing technology we want to use, how we should be integrating those in multimodal systems, or whether we should be building systems in isolation that are interoperable on each of these different types of sensing technologies. So these are some of the challenges that are of interest in addressing in the sensing modalities. The data representation, storing, and sharing formats. This is also of significant interest because this determines how the data will be interoperable, whether the data formats that are used are consistent across different sensing technologies and across multiple device manufacturers and platform manufacturers. So that is why they are trying to look at how to bridge gaps in data standardization, storage, and sharing. And this is one important aspect that this standards development is looking at, because at present there are various formats that are supported by different devices, and there is not much interoperability, although some efforts are there, but they are mostly research-based efforts. But going forward, as you can see that a large number of devices are coming up in the market, and there are more sensing technologies that are going to be made available. So this kind of representation and storing and sharing of data will become more important. So it is therefore important that standards can be built for this kind of data representation, storage, and sharing formats. Even in terms of the end effectors or the actuators, there is no standardization currently. There are different types of actuators. So as I was talking about in the examples that I showed, one of the actuator is a prosthetic arm, right? That artificial limb that the person is wearing, that is an actuator for the EMG pipeline. But there can be other actuators also. There can be exoskeletons, there are VR/AR implementations. Some cases you can say that there are games or there are virtual reality games that are built on this kind of paradigm. So for example, you will think about a particular movement of an object, they will capture your EEG signals and you will move with the object in the gaming environment also. So those kind of VR/AR actuations are also there for virtual reality and augmented reality kind of environments. Neurostimulation is another interesting area where what they are trying to do is they are trying to push back the data that you get from this kind of a pipeline back into the brain by stimulating specific regions. So you might have seen there are some experiments where a person who was physically was not able to visually perceive, their vision was lost, and they used the data from other subjects to train a model and detect different objects. And then what they did was they identified which area that person's brain, in people's brain, get activated when they see those kind of objects. And what they did was they tried to stimulate the brain of the person who was visually impaired, who couldn't see. And he reported that there is some perception of vision, although it is not very clear what they are able to visualize, but still they have that perception of being able to visualize despite having visual disabilities in the occipital region. So there are cases where the entire pipeline is fed back to the brain for stimulation. So that is some cases where neurostimulation is beneficial. And even in this area also, it is very highly experimental, and there are no standards. So this is one area that the standards body wants to go into and work on. Then user needs is another interesting area where we want to work on in terms of development of standards, primarily because there are a lot of applications, right? Some of the applications that I showed you are mostly assistive technology based applications where you are trying to build this system for the users who have physical disabilities. They may not be able to move their arms or limbs, or they may have other restrictions. But even other than these clinical applications, there are a lot of common applications for commercial use. For example, gaming, there are consumer applications, there are entertainment, or we have augmented reality based applications. So other cases, even in fact, including privacy and security as well, where you may want to use these kind of signals for privacy and security monitoring. There are different use cases that are being considered now. So as the number of use cases or the applications of these kind of signals will increase, you will need to have some kind of standardization in the way these signals are processed, these are represented, and how they are used, because that will always help in making these applications interoperable. So That is where, in terms of user needs, we can see that this sort of pipeline for development of standards will focus on. And of course, one of the most important aspects is the performance assessment and benchmarking, where the development of these standards is looking into. So usually you would want that these systems should have low errors, low latencies, and these are what some of the perceptions or the metrics for characterizing the performance of these systems is built on. But going forward, there are more number of applications, more number of signal modalities, and more number of ways in which this kind of data will be used, and therefore the performance metrics or how you will measure these performance of such systems will change. So you will need to look at how to standardize performance assessment and benchmarking, and how you will baseline the performance of the system. So if all of us build different systems, there is no common ground to say which is a better one, right? So there has to be certain baselines in terms of how to evaluate the performance of such systems, and that is where the standards development on performance assessment and benchmarking will be playing a significant role. And on this left here is a set of requirements that have been ranked. So as part of this initiative, a survey was conducted from various users, and this plot is essentially trying to show what is important of different set of applications, right? So consumers have said that, for example, safety, terminology, data ownership, cybersecurity, privacy, performance assessment, data representation, neuroethics, neurostimulation, user requirements, consumer level neurotech, rehabilitation robots and exoskeletons, dual use, and augmented virtual reality. These are some of the parameters that the users were asked to rank in terms of where standards are required, and this is how the distribution of these responses is. So going forward, we see that there will be a lot of work that will go into building these standards, and even IEEE, as I said, has a couple of working groups that are working in this area for building standards to enable interoperability of these systems across different kinds of data, different kinds of applications, and different kinds of performance metrics. So broadly, this is going to be an exciting field. You might have also seen that there are many companies that are working in this area, in fact, including Neuralink and various other companies are working on it, not only from a research point of view, but also for various commercial applications. So this is where, going forward, we see that this will be much talked about space, and there will be a lot of activity in terms of standardization, in terms of machine learning, processing approaches, and the kind of systems that you can build. And we see a lot of work going forward where people in the machine learning, signal processing, neuroscience, and various other communities will contribute towards building these kind of systems. So that is most of the end of my presentation in terms of the slides I had. I basically just wanted to cover some parts of my work and present some future directions, and I hope this has been useful to you. So with that, I'll end my talk and invite any questions that you all may have. So I'll pick up questions from the chat. So neuroimaging domain, if you're in computational neuroscience, if you are interested, there are different ways to start about it. So see, most of us are from an engineering background here, I believe. So your contributions will be from an engineering perspective, right? So one thing that you can do is that there are various courses online that you can look at on neuroscience, on Coursera, on building these kind of systems, and look at literature and try to read papers. That would be one way to start about it. That will sort of help you gain idea about a lot of work that is currently going on. Like the presentation that I showed you, you would have got some idea about what all are the things that are involved in this, and how you can start getting an understanding of this. For example, you have done courses on neuroanatomy and imaging. Those are basically trying to give you an idea from the neurological or the medical perspective. Now, once you have had a basic understanding of the ideas, then you can start looking at specific projects. So you should, even if you go to neurologists in various hospitals, they are also working on a lot of these areas, right? They have patients and they have a lot of cases and a lot of problems that they look at for rehabilitation, for day-to-day assistive technology, for brain stimulation, and a lot of other technologies that they are working on. So you can get in touch with them in their departments. There are professors in academia that can help you at various hospitals, and that will be a starting point where you can work with them on some real problems. Of course, you all come from the engineering background, so you have that computational skill, you have the skill set for signal processing, machine learning, and all those ideas you can implement, and they will provide you data and experiments to conduct with, right? So that is a very ideal use forum in which these cross-domain work can be carried out, and all of you can start working in these kind of areas with clinical professionals. So that is, I think, the best way to go about it. From your perspective as an engineering student, you should have good understanding of machine learning, signal processing, and these techniques. And in terms of trying to do something practical or trying to do some experiments or what sort of applications you can build, you can always work with somebody who is in this domain in terms of either the neurologists or medical help. As people in the medical domain can always look and give you interesting problems to work on, because they both mostly have a medical perspective, but from our side, you can contribute from the entire engineering perspective if you have good amount of data. So I would say that you guys should work on building good skills for machine learning, for signal processing. Anyways, these are going to be used across multiple domains. So it will always be beneficial, even if it is not brain-computer interfaces or neurology. It will generally be very beneficial to start understanding these areas and how algorithms are implemented. And there are a lot of courses on machine learning at this point of time available from various forums, including YouTube videos as well. So always looking at those and learning around the various machine learning techniques will help. And then for practical projects, you can always look at some neurologists. In fact, there is also a forum called as OpenBCI. If you look it up, it's specific to this domain that has a lot of open source contributions, and even in terms of hardware components. So you can look at those forums online and try and see what is of interest, and therefore go ahead from there. So it will be a learning curve. It will not be very obvious to you, but the more you interact and more you try to see social media forums and so on, that will help you get a broader understanding of the picture, and then therefore you can start working in any particular problem that is of interest to you. Look at data sets that are available online. There is a lot of Google data sets has some data sets. In fact, if you look at OpenEEG datasets, you will find a lot of other paradigms also. So you can also do some experiments on your own and try and get a basic understanding, and then you can also go and talk to medical professionals and see what kind of problems they have and what the areas they want your expertise in. So through your departments also, you can collaborate with faculty and try and see what kind of people are working in these areas. So that will be helpful, is what I would say. Any other questions you may have? So I hope, the basic idea that I was trying to say here was to, give you a broad picture of what are the things that are being done in this field. And although all of it may not have been directly relatable because of the kind of difference in perspectives, but like if you have an initial idea, you can always start exploring from this point and go forward and try and see what all is possible, right? So the idea was to look at a few specific applications so that you know what are the important components of these, and accordingly you can work on specific ideas and go and pursue research, or even work in the industry on these ideas. So that is what was my basic objective. So I hope this has been helpful to you.
Sir, it was a wonderful session for us. Thank you, sir, for sparing your precious time for this webinar. And there is a question. Yes, sir. Okay, sir.
Yeah, question that I have answered. Is there anything else? Yeah, I think I have answered most of this. Yeah.
Okay.
So always you are feel free to get in touch, and if you have any questions, you can send them across by email or even other means that you feel suitable. So feel free to talk offline after this as well.
Thank you, sir, for your time. We want just present present small token. Once again. Internet is problematic. Quite. Thank you, sir, for your time. And if anyone have any other question, please ask in the chat box. Sir is still here. We have still time of roughly 10-12 minutes. You can ask your questions right in the chat box.
So as I understand, most of the audience here, they're all first and second year students, or
yes, sir. So
okay, so machine learning achievements is a very broad question. So mostly you will see that in current days and all, there is a lot of talk about artificial intelligence and machine learning. So the broad idea was that if you have enough data for a set of actions or a particular use case that you want to do, you can train a model and you can sort of use a computer or an algorithm to execute that in a scenario. So this was the basic principles with which machine learning started, that you could automate you can sort of realize intelligence through this kind of an approach. So in every domain that you see currently, there will be some or the other application that has benefited with machine learning. So if I talk about healthcare domain, for example, there is automated detection of lesions or cancerous areas. There is diabetic retinopathy. There are many areas where machine learning has been used for improving the quality of medical diagnosis and medical data and implementations for clinicians. So other domains also, if you look at telecom domain, there are people who are working in different problem statements for optimizing telecommunication networks. Even in banking and other areas, you will see that machine learning has been used for various tasks such as fraud detection, for credit risk prediction, and many other challenging problems. If you look at system development or software development, there has been use of machine learning for automatic deployment, automatic testing, so what is called as machine learning operations or MLOps. So these are some examples in system engineering domain. So based on whatever domain that you may talk about, whatever application area, there will definitely be some or the other use cases that have been automated or that have been benefited by machine learning in some sense. So the question will depend on what is the kind of framework we are asking in. But of late, if you look at what all are the emerging applications, what are the emerging areas in which people are working. So one of the areas is definitely this machine learning across the various application domains. So from your viewpoint, I would say that you should use this time to very strongly build up on fundamentals, because machine learning basically is built around maths and statistics and probability, and then of course optimization and various. So it is important to be able to build and understand the basic ideas at this stage, so that later on when you go into applying these techniques for various application problems, it will be easier and you will have a good command over what are the basic ideas of machine learning. So that kind of approach you can adopt at this stage, because you are at a very crucial phase where you have that flexibility to pursue courses and learn new areas. And of course, going forward, there are a lot of areas where you will be able to use machine learning, data science, and all these in some use case or some practical problem or the other. Right? So it's a very interesting area going forward. And if you look at it, the internet, if you look at videos and what, you will be able to see a lot of new applications are coming in, a lot of new companies are coming up that are trying to work in this area. So therefore, in those cases, you will always have good opportunities towards the end of your graduation to look in terms of job opportunities and even research-wise. That is what I would say. Any other question? Okay, biomedical engineering courses which focus on neuroengineering research within them. So neuroengineering, there are biomedical courses that are offered by various institutions. You can focus on either in courses offered by these biomedical departments in various IITs, or there are also specific institutions which work on brain research. So if I were to talk about these BCI systems and brain computer interface systems specifically, one of the key institutions in India is the National Brain Research Center in Manesar, Gurgaon. So NBRC does a lot of pioneering work in understanding brain at various levels. In fact, not only at this EEG level that we are talked about, but even into deeper cognitive processes and neuronal complexities and all those aspects as well. So there are very specific institutions also. You can look up the profiles of the groups. So when you try to decide where you want to go, you should try and look at the profiles of the people there, the faculty there, what they are doing, what are their projects, you will find websites for almost everybody invariably. And you can look up and see what is the area that interests you, right? You can collaborate. They also collaborate with clinical partners as well. So IITs work in conjunction with various government and public hospitals, also private hospitals. So you can look up profiles, you can find details of projects, and in fact, you can also very freely contact and ask them also whether they'll be able to, their work aligns with your area of interest. So that will help you decide which would be a better fit for you based on your aspirations, based on what your interests are, and accordingly you can further pursue collaborations or whatever kind of things will help take forward that project towards the goal. So you may also do it as a formal program. Of course, you have master's programs as you mentioned, but that will also usually involve projects with collaborators, with clinical patients and actual practical applications. So that will be a case where you can go forward based on the study that you could do at this point of time by looking at department websites, contacting faculty, students. That will help you understand this kind of setting better. Okay. So yeah. Anything else you would like to ask?
So probably there is no more questions, I think, and still there is sir's email. You have sir's email. You can mail him. And sir, thank you for sparing your precious time for this webinar. We want just present you a appreciation token from us. And is my screen is visible?
Yeah.
And this is just a small appreciation token for us. Thank you, sir, for accepting our invite also. This was a wonderful webinar for us. I hope many of our students, mostly are from first year and second year, we will be inspired to work in machine learning and neuroscience field. Thank you, sir, for this session.
Thank you. Thanks a lot for inviting me, and I hope this has been useful to you all. Thanks a lot.
Thank you, sir.
Thanks.
Thank you. And everyone, before you leaving, I want to just convey a message that we are IEEE UEM Kolkata Student Branch is started their membership recruitment for 2021 and 2022. So if you students of UEM Kolkata, if you are interested in taking student membership of IEEE, you can contact me directly. Maybe you have probably you have my number in the poster. There is my number. I am from final year EC department, so you can contact me about the membership and all details of IEEE. I will share with you. And if you take membership in this month of September, you will get membership. You will get 50% discount and get the membership service for almost for 16 months. So you will get your membership will valid for up to 2022 December month. If you take your membership, IEEE membership this year. So if any of you is interested to take membership of IEEE and be part of IEEE family and IEEE Student Branch UEM Kolkata, you can contact me anytime. Thank you everyone.
ব্রাদার,
হ্যাঁ বল।
বলছি এটাতে মেম্বারশিপ নিলে কি হবে?
দেখ, IEEE যদি আমি সংক্ষেপে তোকে বলি, আমি IEEE হচ্ছে ইনস্টিটিউট অফ ইলেকট্রিক্যাল অ্যান্ড ইলেকট্রনিক্স ইঞ্জিনিয়ার্স। তবে নামে যাস না, কেননা যার আজকে তোরা ওয়েবিনারে ছিলিস তিনিও IEEEরই সিনিয়র মেম্বার, ঠিক আছে? তো উনিও কম্পিউটার সায়েন্স ব্যাকগ্রাউন্ড থেকে, এবং IEEE একটা প্রফেশনাল টেকনিক্যাল অর্গানাইজেশন, ঠিক আছে? ওয়ার্ল্ডের অন্যতম সবচেয়ে বড় প্রফেশনাল টেকনিক্যাল অর্গানাইজেশন। এখানে সায়েন্টিস্ট, ইঞ্জিনিয়ার্স, রিসার্চার্স, স্টুডেন্টস সব যুক্ত থাকে। তো যারা সমস্ত পেপার, বেশিরভাগ গুরুত্বপূর্ণ ভালো ভালো পেপার, রিসার্চ পেপার IEEE-তে পাবলিশ হয়। যেমন এখানে আমার সাথে মনে হয় এখনো দীপ্তরাজ আছে। দীপ্তরাজের অলরেডি ও মেশিন লার্নিং ফিল্ডে কাজ করে, ও 3rd ইয়ার। তো ওর অলরেডি পেপার পাবলিশ হয়েছে, ও IEEE-তে পাবলিশড অথর। তো পেপার পাবলিকেশন আমাদের ধর কেরিয়ারের জন্য খুবই ইম্পর্টেন্ট। সিভি শুধু বুস্ট আপ করে না, এটা একটা ওপেন করে দেয় আরও ফার্দার যাওয়ার। কেননা আমি বলছি বিটেকের পরেও কিছু কিছু IIT-তে ডিরেক্ট PhD করতে যাওয়া যায়, এমটেকের লাগে না। কিন্তু তার জন্য অনেক টাফ ইন্টারভিউ, টাফ ক্রাইটেরিয়া সমস্ত লাগে। তো সেখানে অবভিয়াসলি তো সিভি, প্রজেক্টস এইগুলোর খুব ইম্পর্টেন্ট হয়, এবং রিসার্চ পেপার। তো যখনই তুই এবং এমটেকের ক্ষেত্রেও আমি বলছি যে IISTর নাম হয়তো শুনেছিস কিনা জানি না, ইন্ডিয়ান ইনস্টিটিউট অফ স্পেস টেকনোলজি, এটা ইসরোর আন্ডারে ডিমড ইউনিভার্সিটি। তারপর IITsগুলো, অনেক IIT-তে তোর ইন্টারভিউ হয়। তো ওখানে তোর সিভি এবং তোর টেকনিক্যাল নলেজটা খুব ইম্পর্টেন্ট পায়। তো যাদের ভালো ভালো প্রজেক্টস রয়েছে করা, এবং প্লেসমেন্টের ক্ষেত্রেও, এটা প্লেসমেন্টের ক্ষেত্রেও ট্রু, কেননা তোদের ফার্স্ট ইয়ার যারা আছিস তারা হয়তো বলে দেবে কলেজ তোদের ফাইনাল ইয়ারের জন্য প্রজেক্ট লাগবে, হয়তো সেকেন্ড ইয়ার থেকে তোদের প্রজেক্ট করতে হবে, বলে দেবে কলেজ। তো ফাইনাল ইয়ারে প্রজেক্ট লাগবে, যেটা না হলে ডিগ্রি দেবে না। তো প্রজেক্ট ভালো করে করতে হবে। তো যাদের রিসার্চ পেপার থাকে, আমাদের কিছুদিন আগেই অ্যালেক্স স্যার আমাদের মেসেজ করে পাঠিয়েছিলেন যে যাদের রিসার্চ পেপার অলরেডি পাবলিশড বা পাবলিশ করবে, তো তাদেরকে ফুল মার্কস দেওয়া হবে ফাইনাল ইয়ার প্রজেক্টে, এবং সেটা যথেষ্ট ইম্পর্টেন্স রয়েছে। তো IEEE হচ্ছে তোর বেস্ট জায়গা নিজের রিসার্চ পেপার পাবলিশ করার। অনেক কনফারেন্স হয়, অনেক ওদের জার্নালস আছে যেখানে তোর পাঠালে পেপার অ্যাকসেপ্ট করলে পাবলিশ হবে। এই ব্যাপারে দীপ্তরাজ আরও ভালো বলতে পারে। আর এছাড়া প্রজেক্টস কর বা রিসার্চ পেপার কর, তোকে অনেক আর্টিকেলস, রিসার্চ পেপারস, জার্নালস, আর্টিকেলস তোকে পড়তে হবে। তো IEEE-তে সেই রিসোর্সটা তোর কাছে, মেম্বার হওয়ার জন্য IEEE সেই রিসোর্সটা তোর কাছে চলে আসবে, ঠিক আছে? স্যার ডক্টর সুমিত সোমান, উনি ওনারও সমস্ত রিসার্চ পেপার, ওনার আমি গুগল স্কলার প্রোফাইলের লিঙ্কটা আমি দিয়ে দিচ্ছি, বা আমাদের ফেসবুক পোস্টে লেখা আছে ওনার গুগল স্কলার প্রোফাইলটা। দেখবি বেশিরভাগ ওনার পেপারই IEEE-তে পাবলিশড, ঠিক আছে? প্রচুর পাবলিকেশন ওনার রয়েছে, অনেক সাইটেশন রয়েছে। তো এরকম সাপোজ কোনো প্রজেক্ট করছিস বা রিসার্চ পেপার করছিস, তখন তো তোদেরকে পড়াশোনা করতেই হবে। আমি শুধু মেশিন লার্নিং জানলাম, কটা মেশিন লার্নিংয়ের প্রসেস জানলাম, তাতে হয় না। এবার তোকে জানতে হবে কি রিলেটেড কাজ হচ্ছে, ঠিক আছে? এবার সেই সাপোজ তোর কাছে একটা আইডিয়া আছে, বা সাপোজ কোনো একটা ফিল্ডে রিসার্চ করতে চাস, তখন তোকে অবভিয়াসলি অনেক রিসার্চ পেপার নিয়ে পড়তে হবে, জানতে হবে, আপডেট হতে হবে যে কী কাজ চলছে প্র্যাকটিক্যাল ফিল্ডে। সো সেইগুলোর জন্য IEEE রয়েছে। এছাড়া যদি বেনিফিটসের দিক থেকে বলিস, আমাদের স্টুডেন্ট ব্রাঞ্চের সমস্ত ওয়েবিনার অ্যাজ UEM স্টুডেন্ট তোরা পার্টিসিপেন্ট হতে পারবি, এবং অন্যান্য আরও আমি কলকাতা সেকশনের আন্ডারে ওড়িশা, বিহার, আসাম মিলিয়ে প্রায় 65টা IEEE স্টুডেন্ট ব্রাঞ্চ রয়েছে, যাদের রেগুলার ওয়েবিনার, ভালো ভালো ওয়েবিনার হয়, ঠিক আছে? তো সেই সমস্ত ভালো ভালো ওয়েবিনারগুলোরও অ্যাটেন্ড করার চান্স পাবি তোরা, এবং IEEEর অনেক ভালো ভালো ওয়েবিনার হয়, সেগুলো চান্স পাবি, কনফারেন্সগুলো অ্যাটেন্ড করার চান্স পাবি, এবং অনেক রিসোর্সেস থাকবে। তো এগুলোর অনেক অ্যাডভান্টেজ রয়েছে, যেগুলো তোর কেরিয়ারে, অর্থাৎ তোর রিসার্চ, প্রজেক্ট, এই সমস্ত দিকে তোকে প্রচুর হেল্প করবে IEEE মেম্বারশিপটা। তো এইটা যদি আমি খুব সংক্ষেপে বলতে চাই, এটা হচ্ছে তোর মানে আমি সংক্ষেপে বললাম। আরেকটা যদি পয়েন্ট আমি বলি, ইম্পর্টেন্ট পয়েন্ট, সেটা হচ্ছে তোদের অনেকে হয়তো কি বলে মেশিন লার্নিংয়ের বা ডেটা সায়েন্সের অলরেডি প্রজেক্ট করছিস বা করবি। যারা করছিস তারা ক্যাগেল ওয়েবসাইটটা তো নিশ্চয়ই জানিস। যারা করিসনি, যারা আজকের ওয়েবিনারের পর ইন্টারেস্টেড হলি একটু মেশিন লার্নিং নিয়ে, ডেটা সায়েন্স নিয়ে, তাদের কাছে এই ডেটা সেট, যে ডেটা সেটগুলোর ওপর কাজ করবি, সেই ডেটা সেটের হচ্ছে খুব একটা ভালো সাইট হচ্ছে ক্যাগেল। তো সেখানে অনেক কম্পিটিশনও হয়। এছাড়া আরেকটা খুব ভালো জায়গা রয়েছে IEEE ডেটা পোর্ট। IEEE ডেটা পোর্টে খুব এক্সক্লুসিভ সমস্ত ভালো ভালো তোর কি বলবো ভালো ভালো তোর ডেটা সেট থাকে, এক্সক্লুসিভ সমস্ত ডেটা সেট, কিছু ওপেন অ্যাক্সেস থাকে, কিছু ক্লোজড অনলি ফর মেম্বার্স। তো ভালো ভালো ডেটা সেট থাকে যেগুলোর প্রচুর মানে কম্পিটিশান হয়, এবং রিয়েল লাইফ ডেটা সেট থাকে, রিসার্চাররা নিজেরা কাজ করছে, তো সেই ওনাদের রিসার্চ রিলেটেড ডেটা সেটগুলো ওনারা পাবলিশ করলেন, এবার সেটায় আমরা স্টুডেন্টরা, রিসার্চাররা পার্টিসিপেট করে, অ্যানালাইজ করে, তার রেজাল্ট সাবমিট করে। তো এই অপরচুনিটিসগুলো এরকম অনেক আছে, ঠিক আছে। এছাড়া নেটওয়ার্কিংয়ের একটা ভালো জায়গা আছে। আজ ডক্টর সুমিত সোমানের মতো একজন ভালো সায়েন্টিস্ট, রিসার্চারের সঙ্গে তোদের কি বলবো, তোরা ওনার ওয়েবিনার শুনতে পেলি, ওনার কন্ট্যাক্ট পেলি, ইমেল পেলি। সো হয়তো মেবি আই হোপ তোদের মধ্যেই কেউ ভালো কাজ করবে, ওনার সাথে কোলাবোরেট করবে, ওনার সাথে হয়তো ভবিষ্যতে ওনার আন্ডারেও বা ওনার সাথেও আরও কাজ করতে পারবে রিসার্চ ফিল্ডে। তো সেটা খুবই ভালো হবে, তাহলে আমি বলবো এই ওয়েবিনার সাকসেসফুল। তো এই ধরনের নেটওয়ার্কিংগুলো খুব কাজে আসে। তো যেগুলোর রেসপেক্টে আমি বলবো IEEE মেম্বারশিপ খুব কাজে দেয়, এবং আরও অনেক বেনিফিটস আছে, সেগুলো পরে মেম্বার হলেও আরও বুঝতে পারবি। তো শর্টে এটা হচ্ছে মেম্বারশিপ বেনিফিটস, আমি যা বললাম, রিসার্চ পেপার, আর্টিকেল, জার্নালস, ম্যাগাজিন, ওয়েবিনার্স, এই হলো গল্প।
বুঝেছি।
আর
বলছি তোমরা কি সাইবার সিকিউরিটি নিয়ে কোনো ওয়েবিনার করবে পরে?
পরিকল্পনা আছে, আমাদের অনেক পরিকল্পনাই রয়েছে। সাইবার সিকিউরিটি নিয়ে রয়েছে, ইসরোর একজন সায়েন্টিস্টকে আনার কথা রয়েছে। কিছুদিন আগেই আমরা আমাদের ফাইনাল ইয়ার মানে পাস আউট হয়ে যাওয়া দাদা, সে এখন ইসরোতে ইন্টার্নশিপ করছে, প্লাস IIST ইন্ডিয়ান ইসরো স্পেস টেকনোলজিতে চান্সও পেয়েছে। সে ইসরোতে ইন্টার্নশিপ করছে অ্যান্টেনা ডিজাইন টিমে। তো এছাড়াও আমার ইসরোর সায়েন্টিস্টকে আনার পরিকল্পনা আছে, আমরা কন্ট্যাক্ট করব খুব জলদি ওনার সাথে। তো আরও অনেক অনেক ভালো ভালো মেশিন লার্নিং, IoT, ডেটা সায়েন্স এবং বিভিন্ন ইন্টারেস্টিং টপিকে আমরা ওয়েবিনার করব। সাইবার সিকিউরিটিও আমাদের পরিকল্পনায় রয়েছে। সো স্টে টিউনড, আমাদের ওয়েবিনারের খবর পেয়ে যাবি, অসুবিধা কিছু নেই। ওকে, আর এই হলো গল্প। আর কেউ যদি ইন্টারেস্টেড থাকিস কন্ট্যাক্ট করতে পারিস আমাদের সাথে, কোনো অসুবিধা নেই। ওকে, তো আজকে আমি এখানেই শেষ করছি ওয়েবিনারটা। থ্যাংক ইউ ফর অল ফর অ্যাটেন্ডিং দিস ওয়েবিনার। আচ্ছা, মেম্বারশিপ কস্ট, হ্যাঁ মেম্বারশিপ কস্ট এখন 50% ডিসকাউন্ট স্টুডেন্টদের জন্য দিচ্ছে। তো 50% ডিসকাউন্টে IEEE স্টুডেন্টদের জন্য দিচ্ছে, তো সেটা 1000 রুপিস মতো লাগে। এবং এই বছর প্রথম তোরা যখন সাপোজ মেম্বারশিপ নিচ্ছিস, তোরা যদি সেপ্টেম্বর মাসে নিস, সেপ্টেম্বর, অক্টোবর, নভেম্বর, ডিসেম্বর, প্লাস পরের বছর জানুয়ারি টু ডিসেম্বর, 16 মান্থস তোরা 1000 রুপিসে মেম্বারশিপ পেয়ে যাবি। তো এটা একটা খুব ভালো অপরচুনিটি। এই টাইমে তোরা 1000 রুপিস IEEE দিয়ে IEEE মেম্বারশিপ নিলি, অলমোস্ট দেড় বছর তোরা ওদের মেম্বারশিপ পেয়ে গেলি। তারপর জানুয়ারি মাসে আবার 2023-এর জানুয়ারিতে তোদের রিনিউ করতে হবে, সেটা আবার 2023-এর ডিসেম্বরে এক্সপায়ার করবে। দিস ইজ দ্য প্রসেস। তো 1000 রুপিস খুব একটা বেশি নয় সত্যি কথা বলতে, কিন্তু অনেক কিছুর অ্যাক্সেস পাবি, অনেক নেটওয়ার্কিং, IEEE-তে অনেক ভলেন্টিয়ারিংয়েরও সুযোগ পাবি। হ্যাঁ, তো সেগুলোর খুব বেনিফিটস থাকবে, সেগুলো যদি ইউজ করিস তোদের খুব সুবিধেই হবে। তো আর আমি আমার নাম্বারটা লিখে দিচ্ছি, হোয়াটসঅ্যাপ নাম্বার এটাই, আমার কলিং নাম্বারও এটাই, ঠিক আছে। তো যদি আর কিছু মেম্বারশিপ নিয়ে জিজ্ঞেস করার থাকে বা IEEE নিয়ে জিজ্ঞেস করার থাকে, আমাকে এই নাম্বারে কল করিস বা হোয়াটসঅ্যাপ করিস, আমি রিপ্লাই করব। ওকে, 78%. ওকে, সো থ্যাংক ইউ এভরিওয়ান, থ্যাংক ইউ অল অফ ইউ. I am ending this meeting now. Thank you, bye.